All right, so my name is Spencer. I'll be working with Brady on this, but he's having some audio problems. So we're going to try and get him through on speakerphone a little later, and hopefully it'll come out clear. But I'm going to go ahead and jump right in. We're going to try and crank this thing out. So I'll go ahead and number one, whenever we have a cross like this, I was always taught to just do separate monohybrid crosses for each individual gene or letter that it's asking for. And so that's all you have to do. And then based on that, you get your fraction. So it'll be a quarter or a half or a quarter for homozygous dominant, heterozygous, and homozygous recessive. And so for A, for the genotype it wants, we'll get 1 in 128. B, we'll get 1 out of 256. C, we get 0 because there's no homozygous recessive D. And then in D, we get 1 out of 32. So that's pretty simple. Number two, fill in the blanks. I'll just try and read through these real quick. Paired homologous chromosomes are sometimes called bivalent or tetrads. The structure that connects the centromere to the microtubules of the meiotic spindle is called a kinetochore. And then C, a diploid fruit fly has 4 pairs of chromosomes. A fruit fly cell in prophase 1 has 16 sister chromatids, while a fruit fly cell in metaphase 2 has 8 sister chromatids. A test cross is a type of genetic cross that can determine if a purple flowered pea plant has a homozygous dominant or heterozygous genotype. E, a non-functional allele is a null allele, and a partially functional allele is a hypomorphic allele. And then F, Mendel's law of independent assortment involves 2 traits, while his law of segregation involves a single trait. All right, so we're going to try and get Brady on, and hopefully you guys can hear him. He's going to do the next 2. Go ahead.
All right, so number 3 was a doozy, just like the rest of you guys said. So T is always going to be homozygous dominant. So the only thing that alternates is the R's and the Y's. So you can have 4 options. You can have homodominant T with big R, big Y, or you can do the T's with small r, big Y, and so on and so forth. They just alternate, and there's 4 options for that. For B, you're going to lose one of those letters for each letter. So you're only going to have 3 letters. So for instance, the first one I have, big T, small r, small y. And they do the same thing where they just rotate through. And then for C, that's going to come from B, and it's going to be 3 plus 3 plus 3 plus 3, and you're going to end up with the number 12. And then for D, the answer that I have is 3. The way I did it was pretty retarded, so I'll just leave it at the answer. And for number 4, dealing with the horse chromosomes and stuff, number A, it says at the end of meiosis 1, the answer is going to be 32. Present during prophase of 1, the answer is also going to be 32. How many sister chromatids are present during prophase 1? That answer is going to be 128. How many sister chromatids are present during metaphase 2? And that answer is going to be 64. And then for E, you're going to have 0 because there's no bivalence during anaphase 2. And kick it back to Spencer.
All right, so number 5 is just like number 1 when we have a tetrahybrid cross. And so all we have to do is 4 individual monohybrid crosses. And so for A, it asks for tall, round, green, and terminal. So that ends up being 9 out of 64, which is 14%. B, it asks for the offspring for a certain genotype, and it ends up being 1 out of 32. C asks for heterozygous for all 4 genes, so it'd just be half times half times half times half, which is 1/16. And then D, it asks for which ones will be dwarf, wrinkled, yellow, and axial, and that'll be 1 out of 64. So that's how we did all our homework problems.